This is a B3 infection and response video looking at the discovery and development of drugs. Now, drug development is a very slow process, okay? Tests and computer models to identify potential new drugs are used first, so we can look in a central kind of resource bank and see what do we know about drugs already, particular drugs that we might be using, how they interact with other drugs, what have other people found out about them in the future. Then it's tested on isolated cells and animals, and that's in the preclinical stage, and then tested on healthy volunteers and then ill patients, and we'll look at why that is shortly. Once that's gone through peer review and it's been okayed for use, then that drug will be licensed for sale to treat diseases, and this can take about a decade. Firstly, we need to look at some origins of natural drugs that we need to be aware of, okay? Traditional drugs can be extracted from plants. Now, most drugs are synthesised by chemists in the pharmaceutical industry. This is because we've got this growing population. We don't really have enough supply in plants, and now we know more about what these drugs kind of consist of. We can synthesise them chemically in the lab. So we need to be able to match up some of these drugs with where they come from. So there is a heart drug called digitalis, okay, and that originates from foxgloves. Now, our digits, our fingers, can be referred to as digits, and our digits, our fingers, would go into a glove. So hopefully we can make that link between digitalis coming from foxglove. Painkillers such as aspirin come from willow, and you'll notice there are 2 L's in painkiller and there's 2 L's in willow, so hopefully that will help us to make that link between painkillers and willow. Penicillin was discovered by Alexander Fleming from the penicillium mold. Okay, so that's the last one that we need to be aware of, but I think we know that from other work we've done, maybe in history as well. Now, drug trials. Really good to watch the drug trial documentary at this point. If you've got time to revise, use that because it covers everything really well. So when there is a drug trial that's being used on the people who are either healthy volunteers or the ill volunteers, we would use what's called a double-blind trial. Now, double-blind means that the people who are part of the experiment, the people who volunteered themselves, the patients, will either be given the real drug or something that looks exactly like the real drug called the placebo. The placebo is a fake drug. It's like a sugar tablet, or if it's an injection, it's a sugar injection. Okay, so some people get the placebo, fake drug, some people get the real drug. Now, the reason why it's called a double-blind trial is because the patients will not know if they have had the placebo or the real drug. The people who created the drug, okay, so the doctors doing the research, the scientists, they will not know either. The problem with the scientists knowing who gets what is they could try and bias the results by encouraging the people who've had the drug that they're feeling better, okay? If there was an independent company, came in, was brought in and paid, regardless of whether the drug proved to work or proved not to work, and they were going to get paid no matter what, then it could become just a blind trial. They've got no interest in whether or not the drug successfully goes through the drug trial or not. So a blind trial is just where the patients don't know if they've had the placebo or the real drug. Okay, so double blind if the people who created the drug are doing the testing. If an independent company are used, then it's just a blind trial. They're allowed to know who's had the placebo and who hasn't. So why do we use a placebo? The word compare. Okay, we use the placebo to compare the results of the people who took the drug and the people who did not take the drug but were in exactly the same environment. Okay, so the placebo is all about comparisons. People in that drug trial group, they may well have similar features, maybe the same age, maybe the same gender, whatever it might be. We like to try and keep as many factors the same as possible. Okay, so tested on people using the placebo, using the drug. The placebo is a fake drug, and if no one knows who's getting what until the end of the trial, it's called a double-blind trial. So why do we test drugs? The first reason why we test the drugs is this safety and toxicity. Okay, does the drug produce any harmful effects? Now that's why we would test on healthy people first, people who don't even have the illness, because if you've already got an illness and then you use a drug test where the drug has safety or side effects issues, then it's going to make you more ill than you already are. So we use healthy people first to test if it is safe and if there are side effects. Once it's gone through the testing with healthy people, we go on to testing the people who actually have the illness. And this is for two reasons. This is to find out the dosage. What is the smallest amount of the drug that's needed in order to effectively treat the condition? Okay, dosage. Then the other use of it is, does the drug work? Efficacy. Okay, does the drug actually treat the condition? Does the person lose the symptoms, lose the disease they've got because of taking the drug? Okay, efficacy is a weird word. Think of it as efficiency. Does the drug do the job or not? And then the dosage. What's the smallest amount I need? I could take 4 paracetamol, but I don't need to, because it's been drug tested to show that 2 paracetamol will do the job. Let's look at the whole drug trial process over 11 years. We've got the preclinical development. Okay, this is where we're looking at computer models first of all, testing just on human cells, maybe testing on animals in this stage as well. Okay, then we go on to the clinical development. So preclinical, no humans involved. Clinical, human testing begins. Okay, and we've got 3 different phases to this. We've got a low dose of the drug tested on healthy volunteers to look at those safety and side effect issues. We've got tested on patients to see the optimum dose, the amount of the dose that's needed with the fewest side effects to get the most beneficial effect. And then we've got phase 3 of the clinical process, which is testing on large numbers of people using the drug and the placebo to do that comparison. How effective is this drug? Once it's gone through the clinical development, so preclinical, testing on animals, testing on human cells, the stuff done in the lab before, computer models. Clinical is about testing on humans, first of all healthy, then ill, and then large numbers of ill using the placebo and the drug. Then we go to the regulatory review stage. Okay, and this is where the findings of the drug trial, what side effects occurred, what benefits came about, it's all reviewed to see, is this going to be a good drug to go on the market. When you read that leaflet inside any kind of medication and it tells you the likelihood of getting different side effects, that's because of the side effects that people had when they did the drug trial. So if it says there's a very unlikely chance of getting whatever it might be, it could be because a few people on the drug trial had that particular side effect. Now that side effect might not be anything to do with the drug. It might just be something they suffered from at that time. But if there's a high chance of getting something, it means that a lot of people on that drug trial had that particular side effect. So we then get to the drug launch. We're at 11 years now, roughly. And it continues to be studied. Doesn't cause... to make sure it doesn't cause the very rare side effects. New formulations such as long-acting tablets may also be developed, so slightly changing the drug. And let's just review that last bit that I was talking about. So we had the double-blind trial. Double-blind trial is if the people who created the drug are testing the drug. And this means that they can't be biased. So all of the drugs will have different codes on them. Some will be placebo, some will be the drug, and nobody knows until the end. So they can't bias the results, trying to encourage people they feel better when they don't. A single blind trial or a blind trial. This is where the people who created the drug are not the people testing it. They've brought in an independent company to test the drug, and they're going to get paid the same fee no matter whether they prove the drug to work or not. So it's just the patients who don't know if they had the placebo or the drug. And then an open trial is where everybody knows. We don't use a placebo. We just say, right, we've got this new drug. You can try it out if you want to. And sometimes people use like asthma drugs and things like that. And they know they're trying out a new drug. Everyone's aware of it. There's no placebo involved. Sometimes the open trial is used as well. I hope you found that useful. Thank you.